Jake there. And Jake is an example, like all the other students. I always say that being in contact with these students always give me a great hope for the future, because they are not just smart, but they are truly kind people. So something that I learned about Jake, and he gave me permission to share this, is that, and that I read on the application, is that he's a creator of an app that you can actually go on the App Store. The name is Sweet Spot. So actually you can go on the App Store and download Jake app. It's actually pretty famous, 5-star, etc. But the reason he designed this app is that not a long time ago he found out that he has diabetes. And instead of being beaten up, he just said, okay, there are not good tools for the people to track what to eat, etc. And so he decided to use his programming skill to build a better tool. So instead of finding himself with all this extra spreadsheet, he found a way to design an app that helped him, but then he's sharing it with the community and working with the community to improve the app. And actually, one day he told me, do you mind if I step out because the TV is coming to interview me? So I was like, say what? So actually, we are going to send out an email, but we are going to see the interview that he got to talk about this app. So it's not only a great app developer, but he also helped one of our new lab here at Max Planck, the Rangaraju lab. So he's going to explain what he did with his, I call it his computer thing, to help the Rangaraju lab.
Hi guys, my name is Jake Thur, and this year I'll be a senior at the Benjamin School. This summer, I had the opportunity of working in the Rangaraju lab, which specializes in neuroenergetics. But what is neuroenergetics? Well, it's an area of neuroscience that investigates how neurons produce and manage the energy that they create and the functions that this energy is used for. So I specifically worked this summer on creating a data analysis software to process massive amounts of data from my mentor Rolene Fan's experiments regarding calcium concentrations in dendritic mitochondria. So even though the brain is only 2% of our body weight, it actually uses almost 20% of our energy. This is because the brain needs a lot of energy to fuel all of its processes, like learning, memory, emotions, and speech. So with all these energy-consuming processes, it clearly makes sense that there's an entire area of neuroscience dedicated to understanding how the brain actually regulates this energy demand. So I'm sure you all remember from biology class that the mitochondria is the powerhouse of the cell because they produce ATP, which is the molecular currency that fuels all of the processes in our cells and our body. From this diagram here, you guys can see that the mitochondria has multiple compartments. So there's the outer mitochondrial membrane here, there's the inner mitochondrial membrane, the space in between those two membranes is called the inner membrane space, and then the inside compartment here is the mitochondrial matrix. And this is actually an image on the right that was taken with a transmission electron microscope, and you can see similar structures from a live cell. So the two compartments that I'll be focusing on today is this outer mitochondrial membrane here, which we abbreviate as the OMM, and the inner mitochondrial matrix. So the mitochondria reacts to different signaling ions, and these signaling ions basically tell the mitochondria to start different functions. So for example, calcium is one of these signaling ions, and an influx of calcium signals for a boost in the production of ATP. And so many studies have shown that a hallmark of neurodegenerative diseases is mitochondrial dysfunction. Specifically in Alzheimer's and Parkinson's, imbalances in calcium concentrations between the cytosol, or the area around the cell, and inside the mitochondria have been observed in patients. So I was testing my software with my mentor Rolene's experiments. And she was investigating the spatial and temporal dynamics of calcium in various compartments of the mitochondria. So she was looking for differences between the calcium outside of the mitochondria versus inside the mitochondria versus on the edge of the mitochondria. So to do this, she would stimulate a spine on a neuron with a 2-photon laser, and that causes an influx of calcium into the mitochondria. And so the GCAM6F protein sensor that she was using to detect this calcium emits fluorescence during these calcium influxes, and we can measure this fluorescence on our confocal microscopes. So this is actually a video from one of our microscopes, and this green fluorescence measures the calcium inside of the mitochondrial matrix here. And also, depending on which experiment you're running, it can also measure the calcium right on the edge of the mitochondria too. And so as you can see, whenever we stimulate this mitochondria, it lights up brighter because there was a calcium influx. And here, this red fluorescence measures the calcium throughout the neuron, specifically the dendrite. So that's why more of it's being lit up, not just in that one area, but same trend. As you light it up, there's calcium influxes and it lights up brighter. So in the Raju lab, a traditional experiment might look something like this. First, we take the images of our samples, which in this case they were cultures, using some form of microscopy. The most common is confocal microscopy, but of course, as the other interns talked about, there's multiple different types and they all serve different purposes. Then we take the images from the experiment and put them through an image processing software called ImageJ. And image processing through ImageJ, when done manually, has to be done with single images at a time. So with an experiment with 300 cells and 2,000 samples and however many conditions, this can be a very tedious step to do by hand. So the ImageJ processing application gives us quantitative data on the fluorescence, and we can save this data in simple Excel spreadsheets. Then we manipulate the data using some type of equations that depend on what we're trying to find in the experiment, and we update those spreadsheets to contain the new data. And finally, we graph this new data using a software called Origin, once again having to individually enter in the data for each cell in order to generate our graphs. And so Origin can give us any graph we want, but very tedious to enter in all the data for an experiment. So my project was basically to code a software that would automate the last 3 steps in this workflow, from the original microscopy images to the final graphing analysis. So I decided to use Python and ImageJ macro language to do this. So of course, I just started to work linearly, and I started with the initial work in ImageJ. So when we take the images on the microscope alternates between the red and the green fluorescence, so that every time we have an image of the calcium in the cytosol, we also have an image of the calcium in the mitochondria to compare it to. So Rolene takes the 2 images of the red and the green, and she stacks them on top of each other in order to draw the regions of interest, or ROIs, that we want to analyze. And that creates this yellow image on the left here. And each circle is one of those regions of interest that she drew. So then Rolene will give me these images, and my algorithm, the first step, is to split the images back into the red and the green channel, producing images like these. And then I take the ROIs that she drew, and I overlay them back onto the 2 new images. Once we have that, we measure the average light intensity inside each of these regions. And we do that for all 120 seconds of the experiment. So my algorithm can create 300 spreadsheets that summarizes almost 100 different cells in about a minute and a half. Now that we have the average light intensities for each region of interest, we need to normalize the data. Normalizing is when we manipulate the data so that everything's based on the same scale, like Ritwik was talking about with his data. And this makes it so that one cell can easily and accurately be compared to another without it depending on how the cell is structured. So if you look at this example spreadsheet on the right, each column here represents the data for one of these circles. So, and each row is a different second during the experiment. So this is the data for the first second of the experiment for this circle, for example. And so we need to manipulate every piece of data in all 300 spreadsheets. And Obviously, this can be very tedious to do by hand. So the algorithm does this part too. And this is also one of our smaller spreadsheets. So some of the spreadsheets can have 30 columns. This one only has 20. It just depends on the cell. So next, we actually have to find where the cell was stimulated in the 120 seconds of the experiment. This can be seen in the peaks of the normalized data because when we stimulate the cell, there should be a calcium influx and therefore a peak in the light intensity. So this is one of the most important parts of the project, and so it's the part of the project that I spent the most time prospecting. So I wrote the code to basically automatically detect the peaks for each region of interest based on parameters that the scientists provide. And the graph here is a visualization of the peaks that the algorithm found, and each number on top is the second in the experiment that the peak occurred. So, for example, this first peak occurred on the 5th second of the experiment, the second peak occurred on the 9th second of the experiment, and so on for the 120 seconds. After all the data has been collected, the scientists can move on to the actual graph analysis algorithm. By manipulating the parameters, the algorithm can give them almost any analysis they want. And because all the data has previously been calculated for the experiment, they can have any graph they want in seconds. These are just some of the hundreds of examples that the software can produce, and as they keep conducting more experiments, the possibilities are really endless. So what are the benefits to using an algorithm like the one I created instead of individual algorithms like ImageJ and Origin? Well, the most apparent one is runtime. The entire process from raw image to graph analysis with my software takes approximately 3 minutes with a relatively large data set of 300 cells. Previously, this could have taken hours or even days for scientists to do this by hand, and the time completely depended on the amount of data they had in their experiment. So scientists can spend more time on their experiments instead of going crazy analyzing them. Another advantage is the adaptability of the program. So just by manipulating the parameters at the top of the file, the scientists can choose between 7 different analysis categories, and there's hundreds of different options in each category to graph. So they never have to look at the code at the bottom of the file or even understand it, and they really just have to pick what they want to analyze with the first 15 variables. I want to say a big thank you to my mentor, Dr. Rulene Fan, who was there the whole time and always asking my biology questions when I didn't understand the experiments. Ilaria Drago for always making sure us interns were at the right place at the right time and constantly rehearsing these presentations. To my lab leader, Dr. Vidya Rangaraju, for making sure I had everything I needed for my project. The whole Rangaraju lab for being so nice and helpful inside the lab. The MPFI supporters for, of course, making this internship possible. It was really amazing. And the other interns for making it so fun.
You said I made you try a lot, but you changed something this morning. So, questions for Jake.
Ritvik. Sorry.
So why the choice of Python over other programs that offer biological image processing packages such as MATLAB?
Yeah, so the question was, why did I use Python instead of something like MATLAB, which already has a lot of the built-in functions that I used. So for me, it's kind of based on preference. My best language is Python, and you can import all of the MATLAB libraries into Python, like Matplotlib and Pylab. So at the end of it, it's mostly the same. MATLAB might have some extra added features, but it was just preference.
Thank you.
Any other question for Jake?
Not a question, but just congratulations on Sweet Spot.
Oh, thank you.
Matthias?
Yeah, congratulations for fantastic software production for the Institute. I'm always looking forward for having this. I'm just wondering, have you talked to one of our other guys that are programming about making a user interface rather than having people change parameters on top of the program? Because that could make it even more useful in the long run.
Yeah, so I actually, that was one of the first things Rolene asked for. And making user interfaces is not super complicated, but again, very tedious. You have to, imagine trying to tell a computer the color, the size, the shape, where it's supposed to be for any size computer. It's extremely tedious. And so I told Rolene that I could do it, no problem, but we decided that there were greater functions that might be more useful for the experiment, and that would kind of be like a last priority.